So the idea is that hybridization means that two populations merge, and that new merged population becomes a new population in its own right, which can subsequently diverge later on. Okay, so in that case, we think of the species relationships as being represented by a network, but gene trees will still be tree-like for this model. So the gene trees will be evolving within these species networks, but they're still traditional phylogenetic trees. So the network is only at the species level. Okay, so here's a simple example of a network on 3 species. So this is a species network. And the idea is that you have these 2 populations that merge, and then they lead to population B. And there's this parameter gamma, and the idea for that parameter is that a lineage that's in this merged population has some probability of having come from the ancestral population on the left, and some probability of coming from the ancestral population on the right. So we let gamma be that probability of going to the left. And 1 minus gamma will be the probability of going to the right. Okay. So if we think about it going to the left, say, then we can imagine that this gene tree came from this 3 taxon species tree where A and B were the most closely related species. If it came from the right, we can think of it coming from this 3 taxon species tree where B and C were the most closely related trees. So there's some probability gamma that this gene tree came from one species tree, and some probability 1 minus gamma that this gene tree came from an alternative species tree. And so we can think of the probability of a gene tree as being a mixture of these 2 different species tree probabilities. Okay, so, right, so we can, there's just sort of an equation for writing the probability of the gene tree conditional on which of these species tree representations make sense, depending on whether it went to the left or the right. And in Meng and Kubatko, who first introduced this approach, these are called parental species trees. Okay, so that approach works well for the type of tree that I showed before. There's a complication that it wasn't really designed to handle, which is if you have 2 lineages that enter a hybridization, a hybridized population or a hybridization node. So in this case, we have 2 lineages sampled from population B or species B. And so when those 2 lineages go up into the hybridized area, there are different possibilities. They can either go to the left or to the right, and they don't have to both do the same thing. So one of them might go to the left and one of them might go to the right. And so the idea that a gene tree can be represented as coming from one of the parental species trees kind of breaks down. And in this situation, you really need to think about the species tree as existing within the network and not really within a particular tree unless we do something else. Okay. All right, so another way or another extension of this is instead of having 2 lineages sampled from population B, we can imagine that the hybridized population diverges. So here it's diverged into populations B and C. And in this case, even if I only have one allele, one lineage sampled per population, it leads to the same kind of problem, which is that the one lineage from B and one lineage from C, if they fail to coalesce in that population that's descended from the hybrid node, I'm going to have 2 lineages entering that hybrid node, and they might go the same way or they might go different ways. Okay, so this kind of case is less straightforward than the kinds of cases that were originally done in that Meng and Kubatko paper. Okay, so what to do with these more complicated networks? So there are a couple of different strategies. There's one that's implemented in Phylogenetic, which is by Loïc Nakle's group. And that represents the network using null trees, and that can handle that. And I have another approach that uses recursion that I did with this, my PhD student, Xiaozhu. That's not quite published yet, but the software is available on GitHub. And the idea is to kind of extend the strategy from the Meng and Kubako paper, and we condition on events that occurred below the hybrid nodes, and we condition on those events, we can simplify the network into a bunch of trees. Okay, so here's sort of an example of a relatively complicated network, since it has two hybridization nodes. So the idea here is we condition on what occurs below the hybridization nodes, so that the B and C lineages, they might or might not coalesce below the hybridization node. If they do coalesce, we can represent that event on the left. And what we've done is collapse one of the edges in the network. We still have a network, but it's slightly simplified because it has one less edge. And on the right, the B and C lineages remain distinct, but now we have this node that has two edges going in and two going out. And so again, we've removed one of the edges from the network. And so those have different weights depending on the probabilities of coalescence events. And the idea is to keep going recursively. So I won't go into all the details for how to do that, but you take these two slightly simplified networks and condition on more events, and that simplifies things. Depending on the events, you might end up with a tree, or you might end up with a simplified network. As long as you have networks, you keep going recursively until you end up with things that are no longer networks. And in this case, you end up with 14 of these sort of parental species trees that you're conditioning on. So it ends up being fairly complicated. So you end up with these mixtures of all of these trees. But the approach is one way of doing it. Okay, so a complication in all the network literature is whether or not networks are identifiable or distinguishable from each other. So I would describe identifiability as meaning you have a large class of possible models, and each of the models in that class gives you a distinct probability distribution. And distinguishability is essentially the same thing, but you're just thinking of maybe a small number of models. Okay, so in this case, we're thinking of two models. So there's a paper that came out last year in PLOS Computational Biology pointing out that these two networks are they're distinct, that they don't mean the same thing biologically, but in some sense they're indistinguishable from most data that you would think of having. Okay, so there's a concept in the network literature of displayed trees. So the idea is a tree is displayed by a network if by removing some of the hybridization edges, you can get that tree out of it. So it's kind of hard to, I don't know if we can use the laser pointer. Okay, there it is. Okay, so for example, if we remove this hybridization edge here, then we're going to end up with this tree over here. If we remove this hybridization edge instead, then we end up with one of these two trees, and we would also have to remove one of these two hybridization edges. So if we remove this hybridization edge and this hybridization edge, then B is most closely related to C, and we end up with this displayed tree. Okay, and it turns out that this network, even though it's distinct, displays exactly the same trees with the same branch lengths as the network on think that these networks lead to different probability distributions, or lead to the same probability distributions on the gene trees. And that's actually true, so I agree with that assessment for this particular case, that they do lead to the same probability distributions on the gene trees. But we can modify the example a little bit. So here I've introduced an extra taxon E, and so we have two species here. And so this is like the case where you might have incomplete lineage sorting, and these two lineages might go different ways. And just by introducing that extra taxon, we still have these two networks displaying exactly the same trees, but it's no longer the case that the networks are indistinguishable. Okay, so the title of the talk is that display trees, well, I can't remember the title exactly, display trees. The idea is that saying that two networks have the same display trees isn't a sufficient condition for saying that the networks are indistinguishable. So this is an example where the two networks display exactly the same trees, but they're distinguishable. Okay. Okay, and it's kind of crucial to the example to make them distinguishable that you have two lineages entering that hybrid node. Okay, and that can happen either because of the hybrid population subsequently dividing, or we could have just sampled two lineages. We could have just sampled two lineages from species B, and that would have worked as well. Okay, so here's another sort of representation of these networks. I like to think of trees and networks using this approach. And anyway, here's a simulation study that this was using Phylogenetic, which shows that yes, in fact, we can do inference to distinguish these trees using multiple alleles. Okay, so the main conclusion was that the equivalence of the display trees isn't sufficient for concluding that you don't have distinguishability. But there's still a lot of open and unsolved problems about whether or not two networks in general are distinguishable. So I'll end there.